So there's one more thing that I'd like to do with you, okay, and I want to just go back and look at the first question, okay, which said we have to show a genetic cross to show how it is possible for both parents who have blood type A, okay, to have a child with blood type O. There is a very specific way, okay, in which we need you to answer these questions, okay, and this actually links to genetics. And it links to doing a monohybrid cross. Okay, and there are certain steps that you have to do and that you have to be aware of for you to capitalize on the number of marks, okay, that you can get in an exam or in a test. So what we did is we already identified that the parents are going to be heterozygous. Okay, so they are both going to be heterozygous if they have blood type A. If they are heterozygous, we've proven now with the Punnett square that it is possible for them to have a child with blood group O. But what I want to run through with you is to show you the formula or the structure in which you have to do this. So, the first thing you are going to say is the P1 generation. Okay, the P1, generation. And under that you are going to write, and you're going to tell me about the genotype, and you are going to tell me about the phenotype of these parents. Okay, so the genotype, we're going to look at that. Okay, and the second one that we're going to look at is the same. It's the heterozygous condition. Okay. The phenotype, however, is blood group A, and that is the same. It's also blood group A. Okay, so the first thing you have to tell me is about the P1 generation. That means the parent generation. And the first things you're going to tell me are the genotypes of these parents as well as the phenotypes of these parents, which in this case happens to be the same. Okay, the genotype is heterozygous and it codes for the same thing, and the phenotype is the same. Both have blood group A. Okay, then what you have to do is you have to tell me that something specific is going to happen over here, okay, which is meiosis, and you can even kind of write it in the center under what you have done over here. Okay, meiosis has to take place, and then what does meiosis make or produce? Meiosis produces gametes. Okay, and if we split that, there and there. If we, via or through meiosis, split the genotypes of these parents, okay, because remember, meiosis ensures that only half of the information from each parent regarding every characteristic is given in a gamete, okay, which is going to ensure that we go from a diploid number to a haploid number, and when the haploid numbers fertilize each other, when a sperm and an egg fertilize, okay, and conception takes place, we are back to a diploid number. So all you need to do is write down what the gametes are going to be, okay, and again, coincidentally in this case, they are going to be the same. You have one dominant gene, okay, because of the A blood type there that you have, the gene for that blood type. You have one recessive gene. And then the parent over there is going to code for the same. One dominant gene, okay, in one egg or sperm, and one recessive gene in the other egg or sperm, depending on if it's male or female. Right. Then what has to happen? Fertilization has to take place. And again, these words you can kind of write under each other, okay. These write on the side so that you make it very clear to the examiner that you have got the correct formula, that you're doing it in the correct steps, and the structure is there. Okay, so your genotype and your phenotype is there, but these two words right in the middle of the page or just in the middle of your work so that they stand out. Now what we can do is the very small quick section that we did now, and that is to show that when fertilization has taken place, okay, when meiosis rather has taken place, then these are the possibilities that can take place with fertilization. Okay, so we can have a child that might have blood group A, and it's the homozygous condition. Then we can have a child over here that could have, again, blood type A, but it is the heterozygous condition. Then over here we can have a child again who is heterozygous, okay, for blood type A. And then over here we can have a child that has blood group O, and the only way we know to have O, because it is the recessive condition, is to have it in a homozygous state. Okay, and then what they might do is they could ask you under that, okay, and what have you, just before we get there, what have you worked out over here? You have worked out the possibilities of the F1 generation. Okay, so this stands for filial. Okay, so the offspring of the parents that you started with. And now you do the same thing. You have F1 generation, you tell me about the genotypes, and you tell me about the phenotypes. Okay, and what we can see is we have to look over here for the genotypes in the F1 generation. That is the first possible genotype in this genetic cross using these parents. Then can you see these two are the same? So the heterozygous condition, okay, and there are two of those. You can only write it down, or you only need to write it down once, because that is the same possibility. And then for the genotypes, we have a third possible genotype, okay, and now here's the thing. The tests or the exams might be structured in one way where it says, write down the genotypic ratio of what the F1 generation are going to show you. And then what we see is, leave a line here. And for yourself, just maybe write in the ratio in any case. Okay, what we see is there is one possibility of that combination. Then that combination, there are two. And then we are back to 1 over here. So your ratio is going to be 1 to 2 to 1. But that is only for the genotype, because now what we see is, for the phenotype, okay, that and that is going to code for blood type A, okay, and this over here is going to code for blood type O. And if they ask you for the ratio over here, then what we're going to have a look at, and you can link it to the genotype, and it's nice. That's why if you've done it this way, you have done all of your working out. You've kind of answered everything that they can ask and that they, might ask. When we look at the phenotypes, we know that that and that are going to code for the same phenotype, even though the genotype is different. They are both going to code for blood type A, which means now the ratio changes from 1 to 2 to 1 to 3 coding for blood group A, and only 1 coding for blood group O. So this is why the genotypes and the phenotypes are so important. They are so closely linked. You have to know what is going on. You have to know that that is the DNA, that's the genetic composition of an organism, whereas this is the physical characteristic that you will see. Okay, and as you can see here, the ratios are different if we're working with the genotype only or if we are working with the phenotype. But if you structure it in this way, okay, you can secure maximum marks. If you are only going to do the Punnett square, okay, you are probably only going to get a maximum of 2 marks. Okay, so these type of questions, these monohybrid cross questions, where they ask you for the P1 generation and then they ask you to show what happens in the F1 generation, this could be something that is anywhere up to about 7 marks, which means you're going to have to go through the whole formula from start, showing me the P1 generation, telling me about their genotypes, their phenotypes. Meiosis and fertilization together are going to give you 1 mark, okay? So you have to have both to get that 1 mark. The fact that you've told me about the gametes can secure you another mark, and if they are correct. And this Punnett square is only 2 marks. Okay, so it's not enough, like we did, okay, previously, where we just quickly for ourselves worked out the genotypes of the F1 generation and of the child. It is not enough to only do this. Okay, to get your full 7 marks, you have to do this from start all the way through, ending with the F1 generation, where you've told me about the genotypes and the phenotypes as well. Sometimes you might have a genotypic ratio, which is the same as the phenotypic ratio, but not all the time. Okay, it could be where in this case you had a 1 to 2 to 1 ratio in the genotype, which then obviously resulted in a very different ratio of 3 to 1 in the phenotype. Okay, and that's why the very first section, okay, of work that we did, the very first page that I did with you, okay, where I highlighted those 6 terms, governs the whole of this chapter of genetics and genetic engineering and blood grouping and things like that. You have got to know what homozygous and heterozygous means. You have got to know what dominance and recessiveness means. You have to know what they are. You have to be able to explain them. And then you also have to know the genotypes and the phenotypes and what they mean. You have to know definitions for them, and you have to be able to link it to this work, because these are the principles that govern all of genetics, and obviously then by default the blood typing. So that brings us to the end of our first session. We're going to take a short break, and when we come back we're going to look at DNA profiling and fingerprinting, and we're going to look at genetic engineering.